Hello, everybody. We are in week 9 of our course. Hard to believe. I know it goes fast and slow, but here we are. So the next few weeks will probably go by a little faster because we can see the end, right? May's not that far around the corner. So we have Chapter 8, Microbial Genetics, and some topics including horizontal and vertical transfer in Chapter 19. So I'll try to make mention of when we're switching over to that chapter material. As always, you will follow along with your study guide. So let's get started. A little bit of review for you. So when we think about genetics, we're thinking about essentially that particular code that is in our DNA. That code essentially is going to code for all of your features, and it also has some redundant areas or areas where it's kind of what we would call junk DNA. But essentially, in each of your cells, and we see here on the lower right-hand side that this is a eukaryotic example. So here's a human, and then of course here's one of our cells, and inside each cell in the nucleus is our entire set of genetic material. That whole library of genetic material we call the genome. Okay, our genome is packaged into replicated. I didn't mean to say replicated into DNA with a double helix. When we look at our cells, they don't always look like these little X's. This is the result of DNA becoming condensed, and what happens is this really stringy form of DNA. Think like yarn. A big old ball of yarn. You know, the classic children's book has a kitten playing with this big old ball of yarn. And essentially, your DNA is in this big old ball of yarn. So when cells are replicating, it would be very difficult to work with that genetic material if it was still in this big ball of yarn. So what happens is that DNA in that form is called chromatin. And the chromatin begins to wind itself up with special proteins we call histones. So essentially, you can imagine if there's a big, huge, wad of yarn on your floor, you might want to start trying to make a big ball. But after you get so big, you may say, well, this ball is too big. I need to start a new one. So this is what happens with the chromatin, is it's essentially wrapped up on one piece of protein, and then it goes on to the next piece of protein and the next piece of protein. Now, in that case, you have essentially these balls of genetic material connected, still connected. It's not like it's isolated balls of yarn or DNA. Instead, it's being wrapped up and condensed and still connected. Then we can see these X-shaped structures, which are what we call the chromosomes. Like I said, this is in a eukaryotic organism. When you take a look at the chromosomes and we begin to unravel it and magnify it, we see that there is that double helix. So the double helix is connected by what we might call the rungs of a ladder. So it looks like a ladder, right? But just twisted. So rungs are stairs, and what we see, or the steps, and what we see in this DNA is that these steps are made up of letters. Now, not really. You wouldn't look at the genetic code and go, oh, there's a C. The letter C is just hanging out there. Instead, that C is essentially an abbreviation for a particular nucleic base called cysteine. T is thymine, A is adenine, and G is guanine. So essentially, the double helix is connected by the pairing of these letters. And they fit together essentially like puzzle pieces. So that's pretty neat. So what we can also see in this figure is that there's the term gene. A gene is a portion of the DNA that is coding for a protein. There's a lot more to that. If you ever go on to a genetics class, you'll learn it. But essentially, there is some start information that says, hey, this is the beginning of this gene. Genes code for proteins, so maybe it's coding for your hair or your fingernails, or enzymes that's produced by your liver, or enzymes produced in your small intestine. So essentially, you have that genetic material there that's coding for that particular protein, and at the end, you have a stop code that says, okay, that's the end of the gene. So we have these different parts of the eukaryotic DNA. So then we think about bacterial chromosomes. So we use an example from one that you can relate to, and now we're going to think about bacterial chromosomes. So what you should remember is that they are circular. The DNA is condensed into a packet using histone-like protein. So it's not the same protein that we use in our eukaryotic cells to wind up the chromatin, but it is similar. And we know that our bacterial chromosomes can come in 1 or 2 or sometimes several different chromosomes. Now in humans, we have 46 total chromosomes. If you take a look at other eukaryotic organisms, it's really dependent on what it is. If you're talking about bees, if you're talking about oak trees, and so on. So hopefully you understand the idea of the genome is the entire genetic code in our cells. Chromosome is essentially the condensed chromatin that forms a distinct, discrete structure that we can see. So we see that in this figure, these X-shaped structures. And then we also see that the chromosomes, if you take a look at them a little closer, or if you take a look at the chromatin a little closer, then you're going to see there are pieces of the genetic code that are coding for a particular gene. Genes code for proteins. So eye color, hair, and hair color, and so on. Okay, so let's take a look at where we find the genome in these 3 different types of things. So I said things because we know that viruses aren't living. So we have eukaryotic organisms. Of course, we have the genetic code, or the genome is inside the nucleus. In a bacterial cell, you might remember that this circular DNA is free in the cytoplasm, so it's not enclosed in a membrane-bound structure like the nucleus. So we essentially have it hanging out freely. And then we also see that, you might recall, that bacteria have this ability to pick up circular pieces of DNA, some like additional pieces of DNA we call plasmids, from other bacteria. So they've got like these like bonus DNA that can help them out. For example, being resistant to an antibiotic, or maybe allowing them to live a little bit better in a particular environment so they can be more successful. Or maybe it doesn't do anything for them, right? They're just carrying around the plasmids, but as soon as that plasmid gets in a cell where it actually gives it some benefit or some disadvantage, then it will have its impact. So let's take a look. Lastly, if we think about the genome of viruses, we're thinking about either some small segments of DNA or small segments of RNA. So now you have an idea of these terms: genome, genes, chromosomes, circular DNA, and where you might find the genomes. Okay, so a little bit more about these plasmids. So plasmids are not present in viruses. They are not present in eukaryotes. And they are these little circular pieces of bonus DNA that's outside of the actual bacterial DNA. So it's not essential for the life of that particular bacterium. So yeah, and you wouldn't think it would be, because it's not its own self DNA. But like I said, it can give an advantage or a disadvantage or have a neutral effect on this bacterium. So there's some examples there. It can make bacteria more virulent, so essentially more nasty, and escape being fought by our vaccines. It can certainly make a bacterium resistant to an antibiotic that has in the past worked, because now it has this piece of information that's making it resistant or not killed by our antibiotic. Okay, so going on a little more deeply into the genomes of viruses. So this is the coronavirus, and you can see its lovely spikes. Essentially, you already know that viruses will have short segments of DNA or RNA, and then they hijack the host cell that they're invading to use the genetic machinery necessary to make copies of itself. So what we know about viruses is they have teeny tiny genomes, right? 10 to 10,000 times smaller than the bacterial genomes. And even the smallest viruses only have 2 genes. But they don't need that. Why do they not need that? Because they're relying on their host cell. So you might remember there are viruses called bacteriophages. Bacteriophages are hijacking bacterial genetic machinery, whereas we think about viruses infecting eukaryotic cells, and in that case, they are hijacking the eukaryotic cellular machinery. So we'll get more into how they do that, but essentially this gives you an idea where you're going to find the genomes and the genetic material in eukaryotic cells, prokaryotic cells, and viruses. Okay, so a little comparison here between eukaryotic cells and prokaryotic cells, and we're just seeing some characteristics like the size of the genome. As you might expect, eukaryotic cells, and we're using human cells as our example, have a whole lot more base pairs. So this is giving you an idea of how many letters are in the entire genome. So we have about 3 billion base pairs, and E. coli has about 5 million base pairs. We can see the chromosomes there. I told you earlier we have 46, and E. coli has just 1 chromosome. And then when we think about the number of genes on those chromosomes, you can see number of genes for human cells on the order of 19 to 20,000 genes coding for all of our proteins, while E. coli has about 4,200. So pretty big difference. So it kind of gives you an idea that the more genetic material we have, likely the more different genes we're going to have, and the more different chromosomes we're going to have, and therefore the more complex we're going to be, right? Let's go back to viruses. Viruses have just enough genetic material to hijack their host cell, right? So they're fairly simplistic, but opportunistic. Okay, so let's take a look. Let me move myself up here. All right, let's take a look a little closer at the arrangement of DNA. And then we want to apply that to the arrangement of RNA. So here in the center of this slide, you see deoxyribonucleic acid. This is our DNA. DNA is made up of a sugar we call deoxyribose. Notice that the deoxyribose looks exactly like ribose, but it alters or it's changed right here. So instead of having an OH group like ribose, it just has a hydrogen there. So it's lacking the oxygen. The other thing that we see is that there's these 3 letters of the DNA code: cysteine, guanine, adenine, and thymine. And it's color-coded here. And what you're going to notice is every time you see a thymine, it's going to be paired with an adenine. Do you see that adenine has essentially 2 components to its structure? Thymine has 1 component, and they connect together perfectly. Okay, so like puzzle pieces. Then we see blue and green is matched up, so we have our cysteine and we have our guanine. So this is the DNA. The double helix of the DNA is made of repeating GATC on one side, and on the other side, it's connected by its complementary base pair. So C's with G's, G's with C's, A's with T's, T's with A's. And remember, the sugar is deoxyribose. So then we take a look at RNA. RNA is single-stranded, all right? So essentially what's going to happen, we'll come back to this, but DNA acts as the ultimate material, the genetic material to make RNA. So we don't need a double helix in RNA. It's not going to stay like this for very long. It's going to be made into proteins. So just remember, DNA makes RNA, RNA makes proteins, and we'll come back to that. The other thing that we see, ribonucleic acid has as its sugar ribose. So remember, we said it has the oxygen-hydrogen groups here. And then we see that the nucleic bases, or the bases making up the RNA, include C, G, A, and instead of a T, we have a U. We call that uracil. So anytime you encounter letters like C, G, A, U, you know you're dealing with RNA. So this is another difference between the DNA structure and the RNA structure. There is a picture in your textbook, so I'm including it here. They're showing a little bit more magnified version of how the nucleic bases are paired. So those puzzle pieces. And then you can see it on a more molecular level. Not that matters. But over here we see our double helix, and then they show us how that looks when we're identifying all the parts of the DNA. You don't really need to know this, but this is from the textbook.